Just 1 second. I'm going to set up the live stream. Hello, everyone. Thank you for joining us today for our ALS Webinar Wednesday series. My name is Alex Yao. I'm the marketing coordinator for ALS North America. I'll be facilitating the webinar today. Just a few helpful notes before we begin. If you're having technical issues with our webinar platform or any questions, type your message or question using the chat function at the bottom of the screen. You can also select the hand icon to notify me of your status so I can assist you. All questions regarding the webinar material will be answered at the end of the presentation. Our presenter today is Sharita Johnson. Sharita Johnson obtained her PhD in organic chemistry, where she worked in the R&D sector, focusing on drug discovery. In particular, her research entailed structure and fragment-based approaches to identify small molecule inhibitors of anthrax, botulinum neurotoxin, various cancer protein targets such as caspase and metalloproteinases, as well as protein targets of the influenza virus. In pursuit of a managerial role, Sharita obtained a project manager position in the analytical chemistry department at ALS Bioscreen and has been working there for 4 years. She's currently the chemistry manager overseeing the analytical chemistry department and is ensuring drug products as well as personal care products are safe for the public. Her career in science has given her experience in every stage of the development cycle, from understanding drug development to clinical trials and now managing the testing of finished products before they're released to the public. Today, Sharita will be discussing method validation of drug substances and drug products. Thanks for taking the time to deliver this presentation. I'll turn it over to you now.
Okay, thank you, Alex, for that welcoming. And I want to say good morning to everyone and thank you for joining the webinar this morning. Like Alex said, my talk is going to be discussing method validation for drug products and drug substances. And the reason why I chose this topic is because at ALS Bioscreen, we do come across where we test multiple types of products for various clients. And I just wanted to reiterate the importance of doing a method validation for drug products and drug substances. But I'm hoping also that I have some members of the audience that are also testing or releasing products such as personal care products, because I think this can also be important for that side of the sector as well. The information and views expressed in this presentation are solely from myself and do not represent any official perspectives of ALS Global Corporation, Bioscreen Testing Services, or any other company or regulatory agency. My talk will entail what is a method validation, why a method validation is important, analytical procedures to be validated, followed by validation parameters and characteristics that need to be considered. And then I will end with revalidations and why that is possible or why that may be needed. Sorry. And then I will allow you guys to ask me any questions that you may have. Validations is the documented act of proving that analytical procedures, processes, equipment, material, activity, or system actually leads to the expected result. Although my talk today will be focusing on method validations, I just want to reiterate that process validation, cleaning validation, equipment validation are all still key components with when you submit your application, your drug application to the FDA. But I will only be focusing on the analytical method validation. So what is a method validation? Method validation is the process of demonstrating that the analytical procedures are suitable for their intended use and capable of giving reproducible and reliable results when used by different analysts. The analytical procedure refers to the way of performing the analysis and the procedures described in detail the steps necessary to perform each analytical test. This may include, but is not limited to, the sample, the reference standard and reagents preparations, use of the apparatus, generation of, say, a calibration curve, or the formulas needed to do the required calculation. So why do we need to validate? Per the FDA guidance document, Analytical Procedures and Method Validation, it describes the method validation process for drug substances and drug products covered by new drug applications, abbreviated new drug applications, supplements to these applications, and drug master files. These applications must contain method validation information to support the adequacy of the analytical procedures. Analytical procedures that are filed in registration documents are then used to ID the drug substance or drug product, quantitate the active pharmaceutical ingredient, determine the level of purity, and then guarantee the overall quality of the product. So basically, the reason why you need to validate is because it's a regulatory requirement. The Code of Federal Regulations, CFR 311-165C, explicitly states that the accuracy, sensitivity, and reproducibility of test methods employed by the firm shall be established and documented. Per 21 CFR 211-194A2, the firm shall indicate the location of data that establish that the methods used in the testing of the sample meet proper standards of accuracy and reliability as applied to the product tested. The suitability of all testing methods used shall be verified under actual conditions of use. Another great reason why you should validate the method is, of course, it's an essential part of good manufacturing practices. So to have quality control and reliable analytical results are necessary to make informed decisions about the quality and the safety of the products in the pharmaceutical industry. And last, method validation is needed because it demonstrates good science and establishes fitness for purpose on the customer's behalf. So then when should you validate your method? As I mentioned in the previous slide, in order to have a new drug or application filed to the FDA, you need to make sure you have the method validated, but you also need to have it validated before the initial use, and routine testing, and when you're transferring the method to another laboratory, and also whenever conditions or parameters within the method has been changed. So how do you validate the method? First, you need to define the analytical requirements and then develop or identify the method. And at this point, this is key stages where before the validation can begin. This is kind of like doing a method feasibility, which allows you to gain familiarity with the method and evaluate the method for attributes such as precision, linearity, accuracy, and specificity, avoiding any protocol acceptance criteria failures. So this is kind of your foundation to kind of develop and make sure the method is as solid as possible. Then you can go forward and plan your validation experiments, perform the experiments, use data to assess fitness for purpose, and then assess whether the analytical requirement was met. If it was met, you do have a method that is validated. If it did not meet those requirements, then you need to go back to the method feasibility stage. I also wanted to mention method verification and method transfers because those are some terminologies that are also used. So I just kind of wanted to explain what they are. Method verification, it verifies that a lab is capable of performing an analytical method reliably and precisely for its intended use, primarily for compendial methods. And then you have method transfers that are performed for validated methods, where a method is being transferred to a new laboratory or a new facility. So then what documentation do you need to submit to the FDA to get your drug out on the market? For method validation, you would need to have a validation protocol. And this document basically specifies how validation of a particular method will be conducted. The protocol should be reviewed and approved. It should also specify the critical parameters and their acceptance criteria. And the validation study is expected to be executed as per the validation protocol. After executing the validation study, you then document this into a validation, the results in a validation report that cross-reference the validation and summarizes all the results obtained from the validation study. This will also contain the well-characterized reference materials with documented purity and should be used throughout the validation study. and the degree of purity is dependent upon the intended use. You will also need to include justifications when you're not using when you're using a non-compendial method. And then, of course, you need to then have a detailed standard test method submitted as well that should provide sufficient information to allow properly trained analysts to perform the analysis in a reliable manner. So as I mentioned previously in the protocol, that you have to have certain acceptance criteria that's specified in that validation protocol. Well, this is called a specification, and a specification is basically a numerical limit or range or other criteria for the test that is described. And it basically establishes the set of criteria to which a drug substance or drug product should conform to be considered acceptable for its intended use. Specifications are critical quality standards that are proposed and justified by the manufacturer and approved by regulatory authorities as conditions of approval. They are also binding standards of the quality between the regulatory agency and the applicant. In general, a drug substance or drug product conforms to the specification when the article is tested according to the listed analytical procedure and the values obtained are within the listed acceptance criteria. Therefore, a pharmaceutical drug product must meet all its specifications throughout its shelf life. So then the next topic I wanted to discuss is what test needs to be performed when you want to validate the method. And for a drug product and drug substance, you have these 4 general tests, which is description, identification, assay, and impurities. However, for drug product, you may have specific tests that need to be validated, and that is dependent upon the formulation of the drug product. So for solid dosage, you will have dissolution, disintegration, tablet hardness, uniformity of dosage, water content, or microbial limit test. Whereas for liquid dosage, you may have all of those as well as pH and reconstitution time, as well as redispersibility and particle size distribution. And then for your parenteral drug products, you have additionally sterility and endotoxin pyrogen testing that would need to be performed. So today I will be focusing more on the general test, and I'm going to describe what each test is. So I didn't list description here because that's basically describing the physical appearance of the drug substance or drug product using either by color, odor, or describing if it's a powder or something like that. But for identification tests, this should be able to discriminate between compounds that are closely related structures, which are likely to be present in your product. This is normally achieved by comparing the sample to a reference standard. And then you have your assay of drug substances and pharmaceutical products. This is basically the measure of the analyte in the sample, which is a quantitative measure, say, of the drug substance in the drug product. The content of impurities and limit tests for impurities can either be a quantitative test or a limit test, which reflects the purity characteristics of the sample as well. So per the ICH guidelines as well as the USP, there are certain characteristics that need to be shown or performed for each of those tests. So I'm going to be talking about ID, assay, and impurities, and these parameters need to be studied in order to have this method considered a valid method. So per the ICH guidelines, you need to look at specificity, linearity and range, accuracy and recovery, precision, detection limit, quantitation limit, and robustness. Whereas the USP guidelines, they added ruggedness and system suitability testing. However, when you are using a USP analytical procedure, since those procedures have already been validated, you have only additional characteristics that will be needed to be tested: the stability of the sample solution and intermediate precision. Okay, so per the ICH guidelines, based on ID and impurity and assay, this is a list of what tests, what parameters need to be tested. And so for identification, it shows that specificity is the only criteria or only parameter that needs to be tested. And then for testing impurities quantitatively, you would need to show accuracy, precision, which includes repeatability and intermediate precision, as well as specificity, quantitation, limit, linearity, and range. And for a limit test, the only parameters that are needed is specificity and detection limit. And then for the assay, you have to look at accuracy as well as precision, including repeatability and intermediate precision, specificity, as well as linearity and range. But you're not limited to those. That's just the requirement that is needed. So now I'm going to go through what each of these parameters are and what type of testing may be needed to be performed so that you can show, say, specificity. So specificity is the ability to assess clearly the analyte in the presence of components which may be expected to be present. This may include impurities, degradants, and matrix. So for identification, you need to ensure the identity of the analyte. And then for assay, for chromatographic procedures, representative chromatograms should be used to demonstrate specificity, and individual components should be appropriately labeled. In case a non-specific assay, such as titration, the combination of the assay and a suitable test for impurities can be used to demonstrate overall specificity. For impurities, if you have impurities that are commercially available, for the assay, you can spike the pure substance, drug substance or drug product with the appropriate level of impurities. And then if impurities are not available, you can undergo forced degradation of the sample, where you store the sample in relevant stress conditions, such as heat, light, humidity, acid-base hydrolysis, and oxidation. Then the two assay results are compared and the impurity profiles are also compared. Next parameter is accuracy, and this is the degree of agreement of test results with the true value or the closeness of the results obtained by the procedure to the true value. Accuracy should be established across the specified range of the analytical procedure. So for assay of drug substance, there are several methods of determining accuracy, such as application of an analytical procedure to an analyte of known purity, comparison of the results of the proposed analytical procedure with those of a second well-characterized procedure, and the accuracy of which is stated and are defined. Also, accuracy may be determined once precision, linearity, and specificity have been established. For a drug product, several methods of determining accuracy are also available. You can apply the analytical procedure to synthetic mixtures of the drug product, components to which known quantities of the drug substance to be analyzed have been added. Accuracy may be inferred once precision, linearity, and specificity have been established. And in cases where it's impossible to obtain samples of all drug product components, it may be acceptable either to add known quantities of the analyte to the drug product or to compare the results obtained from a second well-characterized procedure, the accuracy of which is stated and are defined. When looking at impurities quantitatively, accuracy should assess on samples spiked with known amounts of impurities. In cases where it is impossible to obtain samples of certain impurities and or degradation products, it is considered acceptable to compare results obtained by an independent procedure. And this should be clear how the individual or total impurities are to be determined. So weight by weight or area percent in all cases with respect to the major analytes. For the next parameter, precision, this is an analytical procedure that expresses the closeness of agreement between a series of measurements obtained from multiple sampling of the same homogeneous sample under the prescribed conditions. Precision may be considered at three levels: repeatability, intermediate precision, and reproducibility. The precision of an analytical procedure is usually expressed as the variance, standard deviation, or coefficient of variation of a series of measurements. And the ideal situation is the diagram on the right, lower right side, where it says high accuracy, high precision. That's what you're aiming for. So part of precision is repeatability, and repeatability expresses the precision under the same operating conditions over a short interval of time. Repeatability is also termed intra-assay precision. A minimum of 9 determinations covering the specified range for the procedure where you undergo 3 concentrations in triplicates, or a minimum of 6 determinations at 100% of the test concentration. Intermediate precision expresses within laboratory variations, and it studies the effects of random events on the precision of the analytical procedure. Typical variations to be studied include days, analysts, equipment, etc., and is not considered necessary to study each individually. For reproducibility, this expresses the precision between laboratories. So you have collaborative studies usually applied to standardization of methodology for inclusion of procedures in pharmacopoeias. Validation of tests for assay and for quantitative determination of impurities includes an investigation of precision. So the recommended data to obtain from this study is the standard deviation, relative standard deviation, and confidence interval should be reported for each type of precision investigated. The next one is detection limit, and the detection limit of an individual analytical procedure is the lowest amount of analyte in a sample which can be detected but not necessarily quantitated as an exact value. This can be done either based on visual evaluation, which is primarily for non-instrumental methods, but in some cases it could be used for instrumental methods, such as container closure or something like that. And the detection limit is determined by the analysis of samples with known concentrations of analyte and by establishing the minimum level at which the analyte can be reliably detected. This can also be determined based on signal to noise, and this approach can only be applied to analytical procedures which exhibit baseline noise. Determination of the signal to noise ratio is performed by comparing measured signals from samples with known low concentrations of the analyte with those of blank samples and establishing the minimum concentration at which the analyte can be reliably detected. A signal to noise ratio between 3 or 2 to 1 is generally considered acceptable for estimating the detection limit. The detection limit can also be determined using the standard deviation and the slope of the calibration curve, and that equation is denoted DL equals 3, where sigma is the standard deviation and S is the slope. So similarly, quantitation limit is the lowest amount of analyte in a sample which can be quantitatively determined with suitable precision and accuracy. The quantitation limit is a parameter of quantitative assays for low levels of compounds in sample matrices and is used particularly for determination of impurities and degradation products. This also can be determined based on visual evaluations and determined by the analysis of samples with known concentrations of the analyte and by establishing the minimum level at which the analyte can be quantified with acceptable accuracy and precision. This can also be determined based on the signal to noise approach, and determination of the signal to noise is performed by comparing measured signals from samples with known low concentrations of analyte with those of blank samples and by establishing the minimum concentration at which the analyte can be reliably quantified. A typical signal to noise ratio is 10 to 1, and based on the standard deviation of the response and the slope, the quantitation limit may be expressed as below. The quantitation limit and the method used for determining the quantitation limit should be presented, and the limit should be subsequently validated by the analysis of a suitable number of samples known to be near or prepared at the quantitation limit. Next, you need to perform linearity and range, and linearity is its ability within a given range to obtain test results which are directly proportional to the concentration of the analyte in the sample. It may be demonstrated directly on the drug substance and/or separate weighings of synthetic mixtures of the drug product components using the proposed procedure. For the establishment of linearity, a minimum of 5 concentrations is recommended. And then for range, this is the interval between the upper and lower concentration of the analyte in the sample, for which it has been demonstrated that the analytical procedure has a suitable level of precision, accuracy, and linearity. The specified range is normally derived from linearity studies and depends on the intended application of the procedure. It is established by confirming that the analytical procedure provides an acceptable degree of linearity, accuracy, and precision when applied to samples. The following minimum specified ranges should be considered. So for assay of a drug substance or a finished product, a drug product, normally from 80 to 120% of the test concentration is the range. For content uniformity, covering a minimum of 70 to 130% of the test concentration, unless a wider, more appropriate range based on the nature of the dosage form is justified. For dissolution testing, plus or minus 20% over the specified range. For the determination of an impurity, from the reporting level of an impurity to 120% of the specification. And if the assay and purity are performed together as one test and only 100% standard is used, linearity should cover the range from the reporting level of the impurities to 120% of the assay specification. So next we have robustness, which is the measure of its capacity to remain unaffected by small but considered variations in method parameters and provides an indication of its reliability during normal usage. This depends on the type of procedure that is being studied. However, the reliability of analysis with respect to deliberate variations can typically be stability of analytical solutions or extraction time. And in the case of liquid chromatography, the influence of variations of pH in a mobile phase or variations in the mobile phase composition, temperature, or flow rate. Then for ruggedness, which is only included in the USP and not in the ICH, the ruggedness is the degree of reproducibility of test results obtained by the analysis of the same samples under a variety of conditions, such as different laboratories, different analysts, different instruments, different lots of reagents, different elapsed assay times, different assay temperatures, different days. As I mentioned in the previous slide, when you're performing the monograph test from the USP, it's not a requirement for the ICH that you show stability of a solution. And so based on my opinion, I think the stability of solution just adds another layer of confidence for reproducibility. Establishing the stability of the standard in test solutions at a specified condition, such as room temperature or refrigerated, for a period of at least 48 hours is recommended. So for instance, for related substances, you can prepare your standard or reference solution as per the test method and test solution spike with all known impurities at a specific level at that desired condition. Analyze the standard reference solution and test the spike sample at intervals such as initial 24 and 48 hours against the fresh standard, and then calculate the percent of individual and total impurities. And for assay, you can do something similar to that in which you prepare a standard solution per the test method and test solution in duplicates and keep them at the desired condition and calculate the percent assay of both the standard and test solution against freshly prepared standards. So lastly is the system suitability testing, and this is kind of leaning towards more of HPLC procedures or liquid chromatography procedures. System suitability testing is an integral part of many analytical procedures. The tests are based on the concept that the equipment, electronics, analytical operations, and samples to be analyzed constitute an integral system that can be evaluated as such. And certain parameters that you need to look at are system precision, which is repeated measurements under unchanged condition that show the same results. You want to focus on column efficiency, which is plate count that measures the dispersion of the peak and helps improve the resolution. Then peak tailing, which looks at the symmetry of the peak. Resolution that basically demonstrates how well the two peaks are separated. And then your detection limit, which is usually specified for impurity or degradation products assay. And I know here at BioScreen, we also incorporate a check standard. And although that's not a criteria that was listed in the USP or ICH, the USP guideline, I just wanted to mention here that a check standard does help with the system suitability by checking to make sure that the analyst prepped the standard correctly as well. So basically, you're prepping the standard that is considered the system suitability standard twice, and then you're comparing the weight of the system suitability standard and the weight of the check standard with, if it's an HPLC method with the peak areas, and you're getting a comparison there, and it should be within 1 or plus or minus 0.02 or whatever the criteria you set. So I think that's also a good system suitability test to do as well. I also wanted to mention retention time or relative retention time, because this can also be key throughout the lifetime of your drug product and performing this method. Over time, or if an instrument change, retention time can also shift, and having some sort of criteria in the method would also assist with having a little bit more control and being able to see if something was incorrect or not performed correctly. Okay, so now that we demonstrated that we definitely have a validated method by completing all the tests and performing the key characteristics or parameters that I listed before on the last slide. Analytical method validations is still not a one-time study. It's not where you just, okay, it's validated, I'm done. I want to reiterate that, periodic monitoring and review of the methods and the control labs is just as important. And method revalidation may be needed if any changes in the method or method performance changes with time. And changes can be things such as process for the synthesis of the drug substance, changes in the composition of the finished product, changes in the analytical procedure, transfer of methods from one lab to the other, and then changes in major pieces of equipment, instruments. And the degree of the validation, of course, will depend on the type of changes and its criticality. So things to consider when you're doing a revalidation: you need to ensure that the analytical method maintains its critical performance characteristics such as specificity, sensitivity, precision, all the parameters that we've already kind of went through. The new method, coupled with any additional control measures, improves or is equal to the original method for the intended purpose. And the new procedure is not more susceptible to matrix effect than the original procedure. So you want to kind of package this into a method comparability study where you demonstrate that the changes in the analytical procedure improves or do not significantly change the analytical procedure's characteristics. And you want to employ homogeneous samples from the same batches to be included in the study. And then statistical analysis should be performed to demonstrate the comparability or equivalency of the new method with the existing method. So next, I just kind of want to give you one example that I've seen here at ALS Bioscreen, in which we have a drug product that has been validated and transferred to our laboratory. And the testing that we perform for this drug product is physical appearance, ID, assay, impurities, container closure, uniformity of dosage, pH, reconstitution time, and water content. We perform release testing for this drug product, and one particular lot of the drug product resulted in OOS out of specification result for impurity at a certain relative retention time. And this this method was an HPLC method, so I want to make sure I say that. And so, of course, we opened up an OS investigation, performed reanalysis and retests with different analysts in a different column, and found out that the root cause is the HPLC column. So this column is the same type of column by the same manufacturer, but different lots of that column, but it did not split that impurity peak, which then gave us a false OOS. So this type of example warrants a revalidation. And I just wanted to give this as an example to show that even a column can cause, some small issues, and then you may need to revalidate to check to see if we need to maybe change the type of column that we're using, or maybe adjust the pH a little bit more. So something like this can cause us to want to do a revalidation. Okay, so in summary, method validation is the process used to confirm that the analytical procedures employed for analytical testing are suitable for its intended use, and results obtained demonstrate the quality, reliability, and consistency of the analytical results. FDA, ICH, and USP guidelines provides recommendations for method validations needed for NDAs, ANADs, supplements to these applications, and drug master files. There are key 5 analytical testing procedures that should be included in the validation. Identification, assay, quantitative impurities. Then you have your limit test for impurities. And then I grouped in specific tests because it depends upon what your drug formulation is for the drug product. Then we also went through key validation parameters that need to be carried out for non-compendial procedures such as specificity, linearity, range, precision, detection limit, quantitation limit, robustness, ruggedness, and system suitability testing. Then for compendial procedures, additional parameters such as stability of solution and intermediate precision is recommended. To ensure the quality of the drug product throughout its life cycle, revalidation of methods should be performed when changes occur for the synthetic process of the drug substance, composition of the drug product, analytical procedure, or transferring the procedure to another lab, or if there's any changes in major equipment instruments. So here's a list of all the references that I used to do the presentation. And then I just want to say thank you to you all for listening. I know this was kind of like a basic overview, but I wasn't sure who my audience I was kind of tailoring to, so I kind of kept it as basic as possible. So if you have any questions, just let me know. And thank you to Renil, Wendy, Mitch, and Eileen. I have a great upper management team. They're very supportive, and I just want to say thank you.
Thanks, Sharita. We'll open up the forum for questions now. If you could write out your question using the Q&A function in the toolbar at the bottom of the webinar. Attendees can also see all submitted questions using this window. If you're comfortable speaking, yeah, we'll just use the Q&A function. I'll give everybody a few minutes to submit their questions. Just give everybody a few more moments to type in their questions. I'll review some brief notes. This webinar presentation has been recorded. All participants of this webinar will receive a follow-up email once the recording is available to view. We also post our webinars on our ALS Global website and our ALS YouTube channel in the webinar playlist. I encourage you to find a wide range of video resources and past webinar Wednesday presentations available there. Also, please follow our company page on LinkedIn as we post announcements and registration links to future webinars and other valuable resources and updates. And it doesn't look like anyone has any questions. I guess you covered everything, Sharita. Okay. I just want to thank everybody for attending today. Again, you can contact Sharita for any questions. Her contact details will be in the email sent out along with this recording. I hope everybody enjoys the rest of their week. Thank you.
Thank you.